How's everybody doing today? Good weekend? Yes? Got on top of all your biochemistry? No? Yes? So this is good. I like this. All right. You get an A for the day. Give people a minute or two. Whew, I've been running around. I was afraid I was going to be late to class. And there's nothing worse than the professor being late to class. I think that's a very bad message.
Not that bad.
Not that bad? It is, in my opinion. So I like to be here.
As long as you don't go over after.
I figured I'd hang around till 5:30 tonight and talk. Yes, sir.
What's the cutoff material for the first midterm?
I haven't decided where the cutoff material for the first midterm will be yet. I play it by ear every time. All right. Well, that said, let's get started with stuff. I'm going to finish up saying some general things about amino acids and peptide bonds. That won't take very long here. And then we'll start talking about protein structure up close and personal. And as I've alluded to earlier in the class, there are 4 levels at which we examine protein structure, and we're going to discuss each of those levels. So I know you can't wait for that. Okay. I told you that amino acids are the building blocks of proteins. I told you that there are 20 of them. And you should know that they get joined together in covalent bonds called peptide bonds. A peptide bond is formed by joining the carboxyl of one amino acid, which you can see right down here, to the amine of the next one. And that one you see right here. The resulting bond, which is this guy right here, is a peptide bond. In the process, water is split out, okay? And this covalent bond holds the thing together. This bond is formed inside of a ribosome, which we'll talk about later in the term. Okay? And is essential. Okay? So covalent bonds are, as you recall, very strong bonds. Those bonds hold the amino acids together in a polypeptide chain. Okay. Another depiction of that is shown right here. We see the carboxyl. You see this has been ionized. We see it's an O minus up here, so the proton is already gone off of that carboxyl group. Here's an amine that has an extra proton on it. It's positively charged. What would that tell you about the pH relative to the pKa's of those two groups? Where is it? So it's going to be higher than the carboxyls, and it's going to be lower than the amines, right? That's going to result in the proton coming off the carboxyl, the proton being on the amine. Well, again, you see these guys coming together. There's the peptide bond. Now, one of the things I would say briefly about this bond is this bond gives proteins some properties because of its unusual nature. The unusual nature of this bond is that this bond behaves somewhat like a double bond, not like a single bond. If you recall from organic chemistry, double bonds don't rotate. Single bonds do rotate. Okay? So most of the time, we will treat the peptide bond as if it is a double bond, because it really has a lot of the chemical nature of a double bond, not that of a single bond. The other thing that you see here is that you see structures that arise. We see that we now have what we call an amino terminus, and we also have what we call a carboxyl terminus. Because here was a carboxyl, here was an amine, but now we've got a peptide bond joining these. This is no longer a carboxyl, and this is no longer an amine, at least not a free amine. Okay? So the net result of that is we started with 2 amines and 2 carboxyls, and we end up with 1 amine and 1 carboxyl. If I add another amino acid, I'm still going to have 1 amine and 1 carboxyl out here. So every polypeptide chain will always have a free amine group called the amino terminus and a free carboxyl group called the carboxyl terminus. And of course, these are alpha amine and alpha carboxyl. There's other R groups, and we don't call those that, right? We have 1 free alpha amine, 1 free alpha carboxyl. And that's the amino terminus and the carboxy terminus. That gives what we call a direction to the molecule. We'll see that's important later. Okay. Okay, so now we see a polypeptide, meaning that we have several of these amino acids that have been joined together. You can see where all the peptide bonds are. There we 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 are. Okay. There's our alpha amine, the free alpha amine, so that's the amino terminus, and here's the free alpha carboxyl, and that's the carboxyl terminus. Okay? Now, yes?
So the direction for a change is always going to be from amine to carboxyl?
It's a good question. Generally, I like you to specify that, but the convention is that we make them from left to right as amine to carboxyl. Yes? Okay? Now, I'd like to think about this guy for a second. I'm going to talk very briefly about charge. If you've looked at any of the problems, you may have seen something about what I'm going to tell you here. Okay? We think about, whoa, I want to calculate the charge of this guy at a certain pH. I start thinking, wow, I got an awful lot of things I got to think about, right? I've got alpha amines, I've got alpha carboxyl, I've got R groups, I've got all these various things I have to put together. Well, it turns out that in making a polypeptide, the process actually simplifies. All right? So before, if I were to take and look at every alpha amino and every alpha carboxyl that I started with, what would I have? Well, I would have one alpha amine, one alpha carboxyl, another alpha amine, another alpha carboxyl, another alpha amine, another alpha carboxyl. You see, we start having a lot of those. But, and this is the very good simplification, when we put them in a peptide bond, I told you they no longer act like they're an alpha amine or an alpha carboxyl. Meaning that these guys in here don't get charged. So we don't have to worry about the internal alpha amines and carboxyls for calculating charge because they will not get charged. So if I were to try to calculate the charge on a polypeptide, the way that I would do it is I would have to think of three main things. The alpha amine, because you can see it still has the possibility of being charged. I have to think about the alpha carboxyl. It has the possibility of being charged. And I have to think of any of the R groups that might have charge. Okay? That's the three things I have to think about. I don't have to worry about all these internal former alpha amines and alpha carboxyls because they don't get charged. Well, how will I decide the charge? Well, just like I decided the charge the other day, I compare the pH to the pKa for each group. So let's just do one in class right here. Okay? Let's say that I've got R2 corresponds to an aspartic acid. And let's say that R6 corresponds to a leucine, and R4 corresponds to a glutamine, and R5 corresponds to a valine. Now what I've done is I've picked side chains so that only one of them can ionize. That's an aspartic acid. And now I need to know pKa values. So the pKa values we might say is, well, let's assume for an alpha amine that the pKa value is 9.5. And let's assume for, and I'll give you these numbers on an exam, so you don't have to memorize these. Okay? Let's assume for an alpha carboxyl that the pKa is 2.2. We know it's going to be lower because it's a stronger acid than an alpha amine is. Okay? And let's say for the R group of aspartic acid that the pKa is 4.4. Okay? So we've got 9.5, 4.4, and 2.2. What would the charge on this molecule be at a pH of 3.3? Now, I've given you enough information that you should be able to make that determination. I'm going to take you through it, but it's the kind of thing that I think you should be able to do yourself on an exam. Well, the pH is 3.3. All I have to do is compare the pH value to each pKa value and decide for each one, is the proton on or is the proton off? And then if I know if the proton is on or off, I decide what the charge is, and then I just add the charges up. So there's just a simple set of steps I'm going to go through. At pH 3.3, that pH is more than 1 pH unit below the pKa of the amine group, proton on or off? On, right. pH 3.3 and a pKa of the aspartic acid R group is 4.4. Proton on or off? On, right? pKa of 3.3 and the pH of 3.3 and the pKa of 2.2 for the alpha carboxyl. Is the proton on or off? It's off. So we had on, off. What's the charge of this guy if the proton's on? The alpha amine has a +1. What's the charge of a carboxyl if the proton is on? It's 0. Don't get hung up on that, right? You put a proton on an alpha carboxyl. When you put a proton on a carboxyl, it's a charge of 0. Okay, so we still have an overall charge of +1. What's the charge on a carboxyl if the proton is off? What's the overall charge of the molecule? Bingo. See? Step by step by step. I've got to charge the molecule. Now, you should be able to do that on an exam. Questions about that?
Go over it one more time.
Okay, one more time. I've got 3 groups that can ionize. For each group that can ionize, I need to know what the pKa value is because that's going to determine protons on or protons off. pKa of 9.5, pKa of 4.4, pKa 2.2. Then I told you this whole guy is dissolved in a solution that's got a pH of 3.3. So now I just simply look at the pH versus the pKa and use the rule that I gave you last time, which said that if the pH is more than 1 unit below the pKa, the proton is on. If the pH is more than 1 unit above the pKa, the proton is off. All right, so then I just go through each one. Here's a pKa of 9.5. The pH is 3.3. That means this proton must be on because the pH is more than 1 unit below the pKa. Over here, the pKa was 4.4. The pH is 3.3. That's more than 1 unit below the pKa, so the proton must be on. So the first two, the protons are both on. In the third case, the pH is 3.3, but the pKa was 2.2, so that's more than 1 unit above the pKa. So that proton is off. So now I know on, off, and I have to decide what those groups are. Well, on, we can already see, has a plus charge on it. A carboxyl group with a proton on has a charge of 0. So 1 plus 0 is 1. This guy with a proton off is -1, so the overall charge is 0. Everybody got it? Yes? Clear as mud, right? Okay. Let's see. If you want to see more, come see me. I'll be happy to talk to you. Now, I said earlier that peptide bond behaves kind of like a double bond. It doesn't behave like a single bond. And as a result, this bond doesn't rotate. And that means that this guy lays fairly flat. If I put it on the table, it's going to lie flat. It's not going to twist or turn or do anything. It's just going to lie there. Okay? That plane turns out to be important in terms of determining the overall protein structure. It means that the protein has 1 less degree of freedom than it otherwise would have had. Okay? Let's think about this. All right? So here's our alpha carbon. Here's our alpha carboxyl. Or here was our alpha carboxyl. Here was the alpha amine. And here's the next alpha carbon. Right? If these guys all exist as single bonds, rotate, rotate, rotate. Right? However, if the middle one acts like a double bond, I only have rotate and rotate. So that's what I mean when I say there's 1 less degree of freedom. There's less rotation that can happen when the peptide bond is acting like a double bond. Now that allows us to actually understand structure. I'll say a little bit more about structure in just a bit, but it allows us to do some simplification in understanding structure. And believe me, simplification is good. Okay, this guy right now is very much on my mind because I still have my cold, and histamines are what we produce when we are making snot and all those various things. So I'm just showing you that because I'm thinking about it. No, you don't need to know the structures. Histamine comes from histidine. Okay? Aspartame. You guys know it as nutrasweet. Nutrasweet is made by combining two amino acids together, aspartic acid and phenylalanine. There's a slight chemical modification that's typically done, but it doesn't have to be done. And that's what it looks like. No, you don't need to know the structure. This guy, because it has phenylalanine in it, is something that whenever you read the warning labels on soft drinks, it'll say that phenylketonuria should not take this product. And people who have a genetic disease called phenylketonuria have a hard time breaking down phenylalanine. So if they consume this, and these are in pretty high concentrations inside of a Diet Coke, for example, okay, they could actually do themselves serious harm if they take that. So if they have phenylketonuria, one of the things they do is they really very carefully monitor their diets for how much of this amino acid that they get in there. So aspartame is actually a pretty innocuous artificial sweetener. We'll say more about artificial sweeteners later, unless you're a phenylketonuric, in which case you would really want to avoid that like the plague. I'll talk about that later. I'm not going to talk about that now. And there's phenylketonuria right there. So the problem with people who have phenylketonuria is they lack this enzyme. Okay? And when they lack this enzyme, normally phenylalanine is converted into tyrosine, which is used elsewhere. However, when this enzyme is deficient, phenylalanine accumulates. And as it accumulates, a side reaction produces some of these products over here. And these products are actually fairly toxic. They can damage nerve cells and so forth. So phenylketonuria can actually be managed fairly well with diet. But this is a reason why you don't want to be taking aspartame if you have phenylketonuria. Okay, so much of general things for amino acids. Let's turn our attention to something a little bit more exciting, which is the proteins in 3D. All right. Proteins in 3D, because it's 3 dimensions that everything exists in. Okay? So we have to think about all 3 of those dimensions when we try to understand structure. Well, I said that there are 4 levels of protein structure for us to consider. The first level we call primary. And primary is very simple. We can describe primary structure of a protein in very simple terms. Okay? And I'm going to give you a definition for it. And that definition is the primary structure of a protein corresponds to the sequence of amino acids in it. The sequence of amino acids in it. Why do we worry about the sequence? Well, we'll see why when we talk about the other levels of protein structure. But suffice it to say that a protein that has a sequence starting with methionine and going to alanine and going to aspartic acid is going to have a very different set of properties than a protein that starts with methionine and goes to valine and goes to cysteine, because those R groups are different. So remember that the amino acids differ from each other in the composition of their R groups. If we change the sequence of the proteins, we're going to have changing sequences of R groups. The protein itself is going to be different. The way that proteins differ from each other is by their sequence of amino acids. Okay. If I ask you on an exam what holds together a primary structure of a protein, what would you tell me? Peptide bonds. Peptide bonds are the units that hold together the amino acids in the primary structure of a protein. So primary structure is very, very simple to understand. Okay? Now, I'm going to come back and talk about 5 angles. So first I want to talk about the alpha helix. All right? What's secondary structure? Well, secondary structure is the next higher level of structure that we think about with a protein. It was a structure that was first, I don't want to say discovered, but it actually was discovered. It was described by Linus Pauling, the person for whom this building was named right here, back in the late 1940s. Okay? It's a beautiful structure. Schematically, it looks like this guy here. It's a coil. It's called an alpha helix. And an alpha helix just does this. It's not like a DNA that has a double helix. It's a single helix. It just forms a coil. It's like a curl of your hair. Okay? The curl of your hair actually has a lot of alpha helices in it. Well, why do structures like this arise? The reason that structures like this arise is that the side chains of amino acids can interact with each other. And the carbonyl groups within the... let me back up and say that right. So that the reason we see secondary structure is we have hydrogen bonds that form between the various groups within the polypeptide chain. That's the safest way to say that. Okay? The R groups don't do that much interaction. The coil allows for things to interact. So what we see is, for example, that we have hydrogen bonds between amino acids 4 or 5 amino acids away from each other. That coil brings it around so that now something that's got a carbonyl group up here can have a hydrogen that's pointing at it up here on the next turn of the helix, and that hydrogen bond stabilizes the alpha helix. So hydrogen bonds are helping to stabilize that curly structure. Now, most commonly you're going to see in this class, you're going to see alpha helices looking like this guy on the right, the sort of bluish-gray thing. Because, to be honest with you, when I look at this, and I think most people look at this, it's rather confusing. Each of those red dots there is a hydrogen bond. Okay? But there's just too much information there. My brain doesn't see shape when I look at those balls and sticks. Instead, I use this to guide my eyes, and I see, oh yeah, I see that now. Okay? So an alpha helix is one type of secondary structure. I'm going to define secondary structure for you. And secondary structure arises from interactions between amino acids that are close in primary structure. I'll repeat that. Secondary structure is a structure that arises as a result of interactions between amino acids that are close in primary structure. Now, what does close mean? Well, I said 4 or 5, right? In this class, in essence, we'll say close means less than 10 amino acids away. Something that's further than 10 away, we'll say really isn't very close. Now, the most important thing I've told you about the structure is that there's interactions that give rise to hydrogen bonds, and those hydrogen bonds help to hold this guy together. Okay. There's Linus Pauling's original sketch of the hydrogen bond. How many people of you guys have ever been over to the library and seen the Linus Pauling collection? Yeah, only a couple. You really should go see that, folks. This is the only person on the face of the earth who's won 2 unshared Nobel Prizes. And you can go over and see all of his original stuff in the library. I can guarantee it's a very cool thing to see. You should do that before you graduate. You can see this important drawing here. You actually can see the X-ray image that Watson and Crick looked at and recognized the double helix on. That's over there as well. 2 of the most important things in all of molecular biology are sitting in the Linus Pauling collection. Okay. Well, so you've seen one kind of secondary structure. There's another. Another very common structure, and yes, this was discovered by Linus Pauling as well. And by the way, Linus Pauling won his first Nobel Prize for these structures. Very, very good reason he did. Okay? This is called the beta, what I call the beta strand. Okay? People, you'll frequently hear terms saying beta sheet, beta strand. What's the difference? A beta strand corresponds to one side of this. This is a beta strand. When I start putting them together, I make a sheet of them, and I have a beta sheet. So a beta sheet contains several beta strands. All right? Well, what's the difference between a beta strand and an alpha helix? It's actually quite simple. An alpha helix has a coil, right? You've seen that coil. A beta strand has kinks. Doink, 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 right? It's like the pleats of a curtain. That's what they're trying to depict for you right here. Okay? Doink, 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 right? Now, let's look at the orientation of things here. Look where the R groups are. Look where the next R groups are. In, out, in, out, in, out. Very regular organization that we see of those. Okay? Excuse me. The next question you might ask is, well, what does a protein decide to do? Does it have a beta sheet? A beta strand, or does it have an alpha helix? And the answer is it depends on the sequence of amino acids. The sequence of amino acids will determine what the secondary structure of a protein is. Most proteins will have some regions that are alpha helices and other regions that are beta strands. And the difference will be that some amino acid sequences will favor the coil and some sequences will favor the bends. Okay? All right. There's a sheet. See how they're arranged? Now, I like this figure because it also shows you something. Yes?
So, I mean, going back to that last figure you had, you said that secondary structure is amino acids that are within 10 or so peptide bonds of one another.
But those guys are far away, aren't they? Yeah. So actually, it's a very good question. Let me clarify that since you've asked it. Thank you for asking that. If we look back here, okay, these are actually interactions between chains, but in fact, these guys are interacting within the chain to make that. They're just not shown on here. So this has already interacted with itself to make that beta bend in backbone, and then these things arose between chains afterwards. But it's a very, very good question. Okay. As I said, this structure here shows us how now we see those bigger structures that are out there. So here was kink, 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 right? We start to see a sheet. And these are not uncommon structures that we see in proteins in nature. All right. Well, there's one other thing we have to think about with respect to structure of, secondary structure. And it really is more of a factor for tertiary structure than it is for secondary, but I'll tell you about it here. They're called turns. If we looked at that last figure, let's go back to that last figure. I should have pointed that out. If we look at that last figure that I showed you, look at this. The red is beta strand. What the heck is that? Well, that's a turn. So we have, all right, so if we follow the arrows, we come down. We've got beta strand, we've got turn. We go up, we've got beta strand, we've got turn, okay? So now we start to think about protein structure as arising from secondary structure and turns. Secondary structure and turns. If you want to call turns secondary structure, that's fine in this class. Other places may argue with that. We'll call it turns in this class, okay? The arrangement of these guys now is a function of the turns. And the overall structure of the protein is going to be determined as much by the turns as it is about anything else. The turns are really big variables here. Okay? Let's think about this. Go back to the turns. Well, what are turns? Well, turns arise because of some interactions between amino acids that are about 3 units apart. Here's amino acid number 1, amino acid number 2, amino acid number 3, and amino acid number 4. No, you're not going to draw this. Well, what causes a turn? Well, just like we saw that certain amino acids favored an alpha helix and certain amino acids favored a beta strand, so too do certain amino acids favor turns. And it's here where I'm going to tell you some specifics. Okay? Most common, and there's different types of turns. We're not going to worry too much about that. But one of the amino acids I've described to you already, I've told you enough that you could recognize there's something that's going to be odd about it. One of the amino acids I said didn't have very much flexibility. Anybody remember? Proline. Proline didn't have very much flexibility because, remember, its R group came back and it connected with the alpha amine group. And I said it can't twist like the other ones can. That means it's inflexible. And that means that wherever we have a proline, we have inflexibility. It's not uncommon for proline to be involved in turns. It's not uncommon for proline to be involved in turns. Does that mean that just because we have a proline, we have a turn? No, it does not. But it's not uncommon for proline to be involved in turns. Well, what will determine that? Well, the amino acids around it will play a role in that process. One of the common ones we see is right here. Here's proline. Look at that inflexible guy right here. Look what's next to it. Glycine. Now this surprises people sometimes because what was the property I told you about glycine? It has a hydrogen as its R group. Very good. All right? It has the smallest R group of anything out there. Okay? So why should wouldn't something bulky out here make more sense for a turn? Well, it might, but it turns out that glycine, because it's the most flexible, actually can accommodate that turn very nicely. So when we see a turn, it's not uncommon. We'll see a proline next to a glycine in that turn. I'm going to show you something in a little bit where you're going to see proline involved with glycine, and it's going to make some other structure. Okay. Proline, because of that inflexibility, not uncommonly appears in turns. This is a cool structure. Look at the beta sheet. Okay? This forms what we call a barrel. Okay? It literally is like a barrel. And some enzymes have structures of barrels because what they do is they pass the molecules that are reacting into the barrel to keep them away from water. It's kind of cool. Okay? The macro world structures that we see in terms of barrels or other structures are reflected in the nanoscopic world or the molecular world of molecular biology. Okay. Now, let's see. How do I want to do this? What time is it? Okay. Let's talk briefly about the collagen triple helix. Then I'm going to come back and talk about phi-psi angles. Okay. The most abundant protein in your body is collagen. The most abundant protein in your body is collagen. It is also the glue that sticks you together. It really helps to hold you together. That's what collagen does. Collagen is an example of something we call a fibrous protein. Fibrous. We talk about two main categories of protein. Fibrous is one of them. The other category is called globular. Okay? Now, it's the fibrous ones we're going to focus on for the moment. Fibrous proteins are not the most common proteins among protein structures. Right? Some examples of fibrous proteins. Your hair. Your hair is a fibrous protein. That looks like a fiber, right? It's a fibrous protein. The nails on your hands. Fingernails. Fibrous protein. Other examples. Silk. Spider silk is a fibrous protein. But what's the difference between a fibrous protein and a globular protein? A fibrous protein pretty much has primary structure, it has secondary structure, but it doesn't have much else. We'll see what the much else is when we talk about globular. But when we think of the alpha helix, for example, we think of that coil, 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 coil. It just goes on and on and on and on and on. That's what a fibrous protein does. It could be a beta strand, going on and on and on and on and on. Okay? Silk has the properties it does because those actually form the beta sheets. The strands come together and form sheets and give that sort of smooth, silky feel that we have associated with silk. Well, collagen is a fibrous protein, and it's an interesting fibrous protein because it's not only a fiber, but it's actually a braided fiber of 3 fibers. of 3 helices. Okay? So just like we take a rope or our hair and we braid it, so too does collagen have that property. Okay? It forms what we call the triple helix. There's 3 strands together that hold that guy together. Ah, you stupid thing. Annoying. Okay. Now, let's think about this for a second. How many people in here braid their hair? Who braids their hair? Okay, you have your hair braided. Okay. All right. So if you braid your hair and you don't put a rubber band on the end of it, what happens to your hair? Within a short period of time, it unravels. I used to braid mine, but I don't anymore. I was always afraid it was going to fall out more than it already does. So, all right. Something has to hold it together, right? Well, collagen has a really interesting property, okay? It is stronger if it's held together, but it doesn't have rubber bands to do it. Excuse me. Instead, collagen, after it's made, gets little covalent bonds that hold the individual fibers to each other. So a bond might happen between this blue and this red, between this green and this blue, between this green and this red, etc., all the way up there. Those internal links give collagen tremendously more strength. Okay? The fibers don't come apart. So the fibers are held together by covalent bonds, and I'm going to show you how that happens. It's really cool. It's really interesting. Okay? Let's look first at the sequence of collagen. Whoa, based on what I just told you a minute ago, you're going to say, hey, Kevin, you said putting glycines and prolines together causes bends. This guy shouldn't be anything but bends. And I said, well, no, it doesn't always do that. Collagen's a prime example of where it doesn't do it. And part of the reason that it doesn't do it is, as I said, glycine is very flexible. You flex it one way, you make a turn. You flex it another way, you can make a helix just like this guy. Now, there's some things on here. Glycine, proline, methionine. Glycine, proline, serine. Glycine, proline, right? Then you start seeing some other things. What in the heck is HYP? It's not hype. It stands for hydroxyproline. Remember how I talked about some amino acids on the first day that I said get modified after they're made into proteins? Well, this guy, after this chain got made, an enzyme came along and it put a hydroxyl group on this proline. It put a hydroxyl group on this proline. It put a hydroxyl group on this proline. 2 things. First, there's an essential component for putting that hydroxyl group on there. It's vitamin C. If you don't have vitamin C, you don't put that hydroxyl group on there. That becomes a very important point, which is the next one I want to make, which is that the hydroxyl groups that are on there are the things that make the covalent bonds. Oh. So if I'm vitamin C deficient, I won't put hydroxyls on my prolines. And if I don't put hydroxyls on my prolines, my collagen is weak. And if my collagen is weak, I develop a condition known as scurvy. Okay? Scurvy was originally described in pirates. Great big guys that would go out on the ocean, and when they were out on the ocean, they didn't have refrigeration. They didn't have, shall we say, salads or any fresh vegetables or anything. They had salted meat was all they had to eat on the ship. Well, that meat contains very little vitamin C, and these guys would go out there as big, honking, ugly pirates, and they would come back as little, wimpy guys because their collagen was literally falling apart. Okay? Deficiency of vitamin C will do that. All right? Well, we don't have scurvy occurring for the most part today because people do get enough vitamin C from a variety of sources that happens. But the reason that the collagen falls apart is because that hydroxylation, get over here, that hydroxylation does not occur. Okay. Questions about that? All right. Yeah.
So from that whole diagram, we just needed to know that vitamin C puts energy on proteins. That's what makes it
Yeah.
strong.
Well, anything I said there. But yeah, I mean, what I said was that, of course, that this was very proline rich and it was very glycine rich. And why was this able to form this helix? I said something about that too, right? So again, anything I say is fair game there. Okay. Now, hydroxylysine also is something that's formed. Okay, I didn't talk about that, but that also can contribute to that stability. Hydroxyproline, hydroxylysine. All right, I think I am going to save phi-psi angles until next time because I want to say a little bit about tertiary structure here today while I've got the time to do it. Phi-psi angles I'll save for next time, so I'll come back and talk about that in the next lecture. I talked about fibrous proteins. Okay, so collagen is the poster child for fibrous protein. Keratin, which is the stuff of your hair, is also a fibrous protein. But I said there were also globular proteins. So what's the difference between a fibrous protein and a globular protein? Okay? A globular protein has primary structure, it has secondary structure, and it has tertiary structure. So we're going to have to understand what tertiary structure is in order to understand the difference between a fibrous protein and a globular protein. What you see on the screen is a globular protein known as hemoglobin. Hemoglobin's got some amazing properties, and we're going to talk about them in a lecture coming up. It's one of the most popular lectures I give. People really like to understand how hemoglobin works. You'll be amazed at what hemoglobin can do. If you haven't heard my lecture before, the number of properties in this molecule is incredible. But what's tertiary structure? Okay, let's define it, and then we can think about it. Okay? So the definition of tertiary structure, it's a protein structure that arises from interactions between amino acids that are not close in primary structure. We said secondary, they were close. Tertiary, they're not close. Well, how can that be? Okay. If we think back to the alpha helix, right? We had the alpha helix, we had an amino acid here, we went about 4 amino acids away, and now we can make a hydrogen bond. They had to be close in order to make that hydrogen bond, right? So how can they interact if they're farther away than 10? You could answer that question based on what I've told you already. How could they do that? Turns are the answer. Very good. Okay? So turns put another dimension into this thing. So now I can imagine that I've got an alpha helix, I have a turn, I have an alpha helix, I have a turn, and I have an alpha helix, and now this third alpha helix is close to the first one. If they interact, I have tertiary structure. That's what you see here. That's why it looks kind of globby. Okay? This guy, if we look very carefully, this guy has alpha helix, turn, alpha helix, turn, alpha helix, turn. Okay? And as a result of those turns, sequences that weren't close together now are brought into close proximity. The interactions that happen in tertiary structure are more complicated than those involved in secondary structure. In secondary structure, we pretty much have hydrogen bonds. In tertiary structure, we're not limited to those interactions between the carbonyl and the amine hydrogen, but now we can have interactions between R groups. We can have interactions between all kinds of things. Okay? So let's think about that for a second. Let's take a look at the forces that might be involved in a tertiary structure. I love this figure. This shows what might be a protein that's out there in the real world. It's got an alpha helix, it's got some sheet structure, and it's got a bunch of other just junk out here. Okay? Each of these have various forces that stabilize them. We can see that the helical structure here is stabilized by hydrogen bonds. You know that this guy is stabilized by hydrogen bonds. Do you suppose that hydrogen bonds could exist between these two? Yes. So hydrogen bonds will also help to stabilize tertiary structure. We don't say much about it in this class, but since this figure mentions it, I'll mention it to you as well. Metals can play a role in stabilizing this structure. Metals tend to be positively charged. If we think about a positively charged metal right here, and I have, let's say, a COO minus from aspartic acid and a COO minus from glutamic acid there, we can imagine that this is going to attract those two minus charges and may give some stability to the protein. So a metal or metallic bond can also help to stabilize a tertiary structure. Okay? Let's jump over here. I'm going to come back to these. Let's jump over here and look at this. Okay? Here's a carboxyl, which has a COO minus. I'm sorry, over here, COO minus. And here's an amine group that has an NH3 plus. This is an ionic interaction that can happen, and that can also stabilize a tertiary structure. There's two other forces that can do that, that can help stabilize it. The easiest one to understand is this one right here. Okay? This guy right here is called a disulfide bond. Now, a disulfide bond happens, and I mentioned this to you earlier when I talked about cysteine. If I take cysteine, cysteine has a sulfhydryl group sitting out here. I said it was fairly reactive. You remember that? If I take one cysteine sulfhydryl group over here and I fold that protein so that another one is brought close to it, what will happen is that an oxidation will occur and a disulfide covalent bond will occur between them. That is this bond right here. You can see a sulfur-sulfur bond. It's called a disulfide bond. It's covalent, and as a result, it's a very strong bond. It turns out that disulfide bonds are the strongest bonds that hold together a tertiary structure. The strongest bonds that hold together a tertiary structure. Now, there's one other bond on here I haven't talked about, and I'm going to. And the other bond I want to talk about is called hydrophobic interactions. Hydrophobic. We've talked about hydrophilic. Things that are hydrophilic will make hydrogen bonds with water, right? Well, not everything is hydrophilic. We saw valine, we saw isoleucine, we saw leucine. They have nonpolar side chains, and those nonpolar side chains don't like water. Well, just like if you go to a party and there's somebody you really don't like, but there's somebody that you really do like, you're going to hang around with the person you really do like and ignore the person that you really don't like. You're exhibiting a hydrophobic behavior. All right? These side chains of these amino acids that don't like water prefer to associate with each other and not with water. There's an attraction for them. That helps to stabilize the tertiary structure of a protein. Now, here's the real kicker with that. And this is kind of cool. Proteins fold to make this. You can see the folding that's happened to get to this point. Proteins that are dissolved in water will fold themselves so that the hydrophobic amino acids are on the inside of the protein and the hydrophilic ones are on the outside of the protein. Why? Because the hydrophobics like each other, and hiding on the inside is the best place to be. Okay? So hydrophobic amino acids will be on the inside of a folded protein that's dissolved in water. You know when you cook an egg, do you know what you're doing? You're taking albumin, which is really nicely dissolved in water. It's viscous, right? It's very thick. You're boiling it, and when you're boiling a protein, you're going to start disrupting bonds, right? You're going to break hydrogen bonds. You're going to break ionic bonds. You're not going to break covalent bonds, by the way, by heating it. Okay? But you're going to break hydrogen bonds. You're going to break these other bonds. And now those things that were very much hydrophobic amino acids on the inside of the protein, guess where they're going to be? On the outside of the protein. And do they like water? No. They precipitate. That's why the egg goes from being a liquid to being a scrambled egg. You've denatured the protein in the egg. Okay? Protein structure tells you some cool stuff. Can you put it back? No. Most proteins you can't put back. And no, you can't unscramble an egg. It'd be a concept for you nutrition majors to think about, right? Unscrambling the egg. Okay. Questions about that? Should we sing a song for the day?
Yes.
All right. This one is to the tune of "Oh Little Town of Bethlehem." And it says the instructor sing, but please, everybody sing it with me, okay? Oh, little protein molecule, you're lovely and serene, with 20 zwitter ions like cysteine and alanine. Your secondary structure has pitches and repeats. I can't hear you. Arranged in alpha helices and beta pleated sheets. The Ramachandran plots are predictions made to try to tell the structures you can have for angles phi and psi. We'll talk about that next time. And tertiary structure gives polypeptide zing, because of magic that occurs in protein folding. A folded enzyme's active and starts to catalyze when activators bind into its allosteric sites. We'll talk about that too. Some other mechanisms control the enzyme rates by regulating synthesis and placement of phosphates. And all the regulation that's found inside of cells reminds the students learning it of pathways straight from hell. You can relate to that. So here's how to remember the phosphate strategies. They turn the GPBs to A's and GSAs to B's. A lot of things you guys will be learning very soon. Okay, see you tomorrow.
That might be a stupid question, but we'll find out. On this one, when you were showing it earlier, we were just calculating the charge. I understood that the H plus is on, H plus is on. That's a +1, that's a -1, and so and then it gives you overall 0.
Yeah.
But you set up here the carboxyl group.
Yeah.
But it was an aspartic acid.
Yeah. So you put a proton on a carboxyl group, what do you get?
Aspartic acid?
Any carboxyl group, charge is 0.
Oh, okay. So that's still a carboxyl group.
Yeah. Okay. Sure.
Why do the peptide bonds behave as double bonds?
Why do they behave as double bonds? Because they have electronic resonance with the double bonded oxygen, which means that there's extra electrons in that oxygen, and they sort of flow down into the carbon nitrogen bond, and that gives it the character of a double bond.
Okay.
It's called resonance, is the phenomenon. Yes?
The metal ion and the tide bend. That's why we require iron in hemoglobin.
Is that why you require iron in hemoglobin? No. That metal ion that's there, hemoglobin, the iron in hemoglobin has nothing to do with the, or very little to do with the tertiary structure of hemoglobin, as we will see. But we need iron to carry oxygen. That's the primary function of iron. Primary function of iron in there.
What about chlorophyll?